the absence of an actual class period where we can do an exam review, what I'm going to do is just kind of take the review sheet that I have, one that's the condensed one, and the other that is the very long original one that was posted online that also has with it things like these tables that go through the groups of bacteria and some of the characteristics of them that I talked about in the little previous lecture. And I'm going to kind of go through it just now as quick as I can, but as to be as inclusive as I can to give you an idea of what to really focus on as you're studying or preparing for the exam. So I'll just kind of go through them, and I'll write down some things that you should take note of, or just kind of point some things out. So first part, I have overview and history. I went through a little bit of the history of some of the scientists who were pioneers. We're going to talk about a few more of them later on in the course. So I would say listed Van Leeuwenhoek and Robert Hooke are both credited with early creation of the microscope. So what you should be able to do is, what's the difference between them? Who came first, for example? Who invented the microscope of the two of them? Some people get this wrong, even in publications, but since Leeuwenhoek read Hooke's Micrographia, obviously Hooke had published that and used the microscope before Van Leeuwenhoek. So Hooke was the first one. Van Leeuwenhoek is noted because of his use of the microscope to see bacteria. All right, so Van Leeuwenhoek saw bacteria first, but it was Hooke who used the microscope first to see other things and actually coined the term cells. The term cell comes from Hooke. I mentioned a couple other people. They're on the list. Pasteur, who he said he's known for many things, and he'll come up again in the course several different times. But he was known for disproving the spontaneous generation, so that inanimate thing or a non-living thing could sprout life. And essentially his experimentation with the swan neck flask is what demonstrated that in a way that was accepted by other scientists, so they didn't anymore follow that particular theory, and led to the study then of bacteria. More so, even though they didn't necessarily know a lot about it at the time. Robert Koch then continues that with his postulates, which I said we haven't focused on intensely yet, but we will so as we do this in the lab. So this is where you you find an organism that has an infection, you try to isolate the infectious agent, you then infect a healthy individual with that agent, and then try to re-isolate the same exact organism and compare it to what you originally isolated. And if you can do that, then you've found what causes the disease. So his investigations have done that. Semmelweis I put on here just because we did the hand washing lab, and so he was one of the first ones to really start to stress that contagious organisms of some kind can be spread through touch, essentially, that there are small microscopic organisms that we can't see that cause disease, and that those diseases can be spread around us. And it's a good idea, say, for people who work in a hospital to wash their hands before they put them in a person during surgery, which they were not doing at the time. So that's sort of significant. So those would be more probably in a matching section, some of the names of these people. Microscopes. And we did a little bit of this in lab. So now the Tuesday people, that I would have gone through a quick lecture during the lab period. So the main terms here for microscopes are magnification and resolution. So magnification is just how many times larger we see things. The individual objectives magnify it, and then the eyepiece magnifies it. So the total magnification is the combination of the two. Now, as you increase magnification, you need more light. So that's one thing that happens. And also the resolution can potentially decrease unless you have a way of counteracting what's going on with the refraction of light. So that's where oil immersion comes in. The oil immersion focuses the light beam, which then helps us keep the image brighter and aids us in focusing on it with better resolution. All right, so resolution is our ability to tell the difference between 2 points, that they are actually 2 points as opposed to just one big blurry point. And so that's kind of resolution. The different types of microscopes, and the main thing is we're using a bright field light microscope. So most all the microscopes on the list are light microscopes. They use a light source, like a light bulb, and that the field that you look at is bright. It's full of light. The organisms, the things that you look at are stained or dark in that. A dark field, in contrast, is a black background, and the things that you're looking at appear light. That would be like this area here would be like a dark field kind of simulation. Then we start to get into cell structure. So this is going to be really the main, one of the main things on the exam of a lot of different questions, probably relating to some aspect of cell structure. So I would start off really focusing on the cell wall. All right, so make sure you know the differences between, well, first off, what is a cell wall? Be able to sketch the peptidoglycan overall structure. And what I mean by sketch it, I mean something just more like, NAM, NAG, NAM, NAG. I'm just going to do a little section here. NAM. It's become have to do quite a bit of it because it's not a little tiny thing. It's kind of a massive structure. So there are bonds, glycosidic links between all the sugars. So these are our sugar molecules, and then we have the tetrapeptide cross bridges that occur. Okay, so peptide cross bridge. So we have the peptide glycan. These are sugars, and that is the main structure, you break in as simple as I can quickly do it, of the cell wall for bacteria. Now, for the gram-positive bacteria, it's the peptidoglycan, and it's very thick. There are many layers of the sugars. For the gram-negative bacteria, again, it's a very thin layer of sugars. And then on top of that, they have an additional second phospholipid bilayer. So I would be able to, do that type of thing in a really simple sketch as well, where you're saying, let me do something like this. So we're going to say this is cell membrane. And so this is the cell's interior. So these X's are, that's the cytoplasm, say, of the cell. And then for the gram-negatives, there's an additional membrane. So the gram-positive has the membrane, the peptidoglycan layer. The gram-negative has the membrane, the peptidoglycan layer, which is really thin, and then a second phospholipid bilayer, a second membrane that's outside. Some of the other things then. We have, for the gram-positive bacteria, something unique. They have things called lipoteichoic acids. The lipoteichoic acids, that's unique for gram-positive. And then they have just plain teichoic, which are just embedded in the peptidoglycan layer. They're sticking out. Gram-negative, they don't have those. But they do have something different. So let me try to get a different color. So they have the thing we refer to as Lps Lipopolysaccharides. These are the things that are endotoxins. So in many of the gram-negative bacteria, they are the things that cause an irritation or even disease, right, in some organisms. They don't have the teichoic acids. Gram-positive, we said some of them can have an extra protein layer, outside this as well. Here, the periplasm, so that's the space, between these two. Make sure you know that term. Periplasm is the space, say, between the membrane here and the peptidoglycan, but it's kind of small technically in this group. Here it kind of expands. It's a lot bigger, so they have a much bigger periplasm. That would be the space also where in the spirochetes where they have their flagella, the flagella that wraps around those elongated corkscrew-like cells exists actually in the periplasm. And we'll talk about that with active transport pumps like in the mitochondria that actually pump protons. They'll pump it into that periplasmic space. So that periplasmic space is important. Make sure you know that. As we continue to add on to the cell. Outside of the cell, or both of these, okay, we can get other sugars, right, that can be to the outside. And so these other sugars are part of what we call the glycocalyx generically, and it can form a slime layer, which is loose, or a capsule if it's organized. Okay, that is outside of the cell wall. Now they're also for a group of gram-positive bacteria that have mycolic acids and substances that are waxy that are outside this area, and they prevent the uptake of the dyes for the gram stain. So there's another technique that we'll go through in lab that helps you. Let's look at those. As we're just looking at structure of the cells themselves, then we have to look at all the other things associated with the cells. So things like extensions then of the cell that stick out, like the pili, that are going to connect, potentially one cell to another cell, or that allow cells to move in a different way than a flagella, and the fimbriae. So one of the differences here between them are going to be the fimbriae are usually numerous, or sort of hairy-like, or pili are usually singular. There are few of these in number. Pili can also be used for the exchange of genetic material, which is something we get into later. Fimbriae do not. They're not used for exchange of genetic material. Then we have the flagella, so make sure you get the basics of the flagella structure. And again, in relation to the membrane, so what we're going to have is always, for both of them, on the inside, okay, of the cell. On the cytoplasmic side, we're going to have proteins that are building, this flagella. They're going to be motor proteins and rings, and ring proteins. So the rings are actually going to be, round, wheel-like structures. Now, in the gram-positive bacteria, they have fewer layers. So the flagella is less complex. Whereas in the gram-negative bacteria, they can have a variety of proteins associated with these different layers. So that's just one of the differences. But you're not going to be responsible for sketching the specific differences between the two. Just know that each of the layers may have different proteins that anchor them. So the gram-negative flagella would be then more complex. That's the way I'm looking at it. Both of them would then have like a hook-like structure, which is a protein. All right, and then the actual flagellin proteins themselves, all right, they can come out. Now, the arrangement of the flagella, they can have a single flagella, they can have two flagella, they can be a tuft of flagella, they could cover the whole cell. So this is, again, totally different kind of structure than the pili and fimbriae. So just know, the basics of it, the differences. If you're sketching it, it's a super simple kind of sketch with the label. So say just something like I just here, just a little curved structure saying, well, that's the hook, and this part would be the flagella. This would be motor proteins. And then if there was a little ring protein, that's what the motor proteins spin. So they spin the ring, and then this hook turns around, and it actually makes the flagella spin in a corkscrew-like pattern. And that would be pretty much it. So if you're describing how a flagella works, that would be it. ATP is used for energy, powers the motor protein, spins a ring, which spins the hook, which spins the flagella, and it allows propulsion of a bacterial cell, which is also very different than a flagella in a eukaryotic cell, which I'm not going to get into right now, but it is different. So what else about so internal structures? So we know that there is no nucleus. The nucleoid region is where the DNA is held. There can be inclusions. There can be little concentrated areas of material inside the cell, but there are not intracellular structures made up of membranes inside any of these cells. As we go outside the cells, again, into maybe a grouping of cells, we can get some more characteristics. So we can have the actual shape of the cell itself, so the cocci, the round ones, and the more rod-shaped bacilli. And then know the terms where if you get chains together, you can have the streptococci. Otherwise, they're in clusters or groups, which are staphylococci. Bacilli can also have strepto. They can be in chains, as well. So just know that term: strepto versus staphylo, cocci versus bacilli, and then the spirilli, or the corkscrew-shaped ones. So those terms, that part is fairly important, and probably going to weigh heavily, into the exam on all the some of the generic aspects of the cell wall. The details of the cell wall itself, This is the one area that's going to really have the most detail, probably, like depth to it. And then all the other things associated with it are going to be kind of light. They're not going to have a lot of depth to them, but there are a lot of other things. So you need to know the terms, essentially the definitions. So that's kind of where I would start, essentially, in studying is just terms and definitions. A lot of the questions will be definition based. For one, that's all the question will be is, do you know the definition of this term? And that's it. Others, if the question is a broader question, within it there'll be a term. And if you don't know that term, then you can't even attempt to answer the question because you're going to need to know it. So the terminology is the key place to start. Like I said, and all these things don't have a great level of depth to them that we need to know. They do. There's all the chemistry, there's all these other things you can go into, but we have too many topics to cover to really go into depth in many of them. So there's just a few that we do, and the cell wall is one. So don't skip out on that one. Make sure you go into it in detail. As far as then we went into actual then classification of organisms, which I kind of went through the basic layout of it on the previous little video that I did. But essentially, the other thing that you need to know are, again, some of the other terms, and these have a little bit of process to them and some rules, has to do with the techniques used in classification. Right, so I said the 16S ribosomal RNA, or we also call it the small subunit ribosomal RNA. The sequencing of the genes within that are what are primarily used for classification. So a question then related to that would be, why? Why this one? So I might say, give me 3 reasons why that is a good gene to use. So you should know that one is it's highly evolutionarily conserved, essentially meaning that all cells have the ribosomes and all cells have the small ribosomal subunit. Okay, so as far as all the housekeeping genes go, the genes that the cells have that they're using all the time to stay alive and do all their basic processes, this is one of them. It's a reasonable size as well. What I had read recently is actually the large ribosomal subunit is actually better for looking at evolutionary relationships. It's better than the small subunit. It gives much more reliable data on how close organisms are to one another. There are a lot more errors actually found with the small subunit, but the size of it is far more reasonable to work with when you're working with a lot of organisms and handling a lot of samples. So it's a reasonable size. The rate of mutation in it is known and is set at a rate that is not too fast essentially or too slow. So if there's too much mutation, it's hard to interpret the data. If there's not really any mutation or much change over time, again, then you're not going to see any difference between organisms. So we know the standard sort of rate of mutation or change, for this gene, it can be used for eukaryotes as well. Eukaryotes will use other things, but it's really primarily for the bacteria and the archaea, which we also got into a little bit. The 16S ribosomal RNA can be used among then different domains of life as well. And it was one of the things that was actually used to separate the domain level, and it's something now that we focus in on at the smaller level. Well, so that's sequencing genes in a specific gene here. Now using more genes is typically better. So instead of just using, so we call this a polyphasic multiple genes and techniques. So this one's important, but instead of, if we just use it, like I just said, there can be some errors. There could be some problems with it. So picking out other housekeeping genes to use, that's many phases of analysis. We start to build cladograms and diagrams, right? I did a little bit of this in class. And so these could be different genes, so at each of these branching points that we're using to identify or classify organisms and see, who's more closely related to one another. So like, let's say I had a drawing like this, A, B, C, D, right? And it was just a question. It said, who more closely related. Look at the drawing, and just right here, C and D are at the end together, so obviously they're more closely related. Or I could say, are C and D more closely related than A and D? Yes, obviously they're just more close together. That might be true if we use, say, one method of analysis, but as we use multiples, it may hold true or it may be changing all the time, which is why we use different things. Other things we could do are the DNA hybridization, which we talked about in class. So hybridization is how we cut up the DNA into little pieces. So be able to explain this a little bit and potentially answer some questions about it. So this is where we just take the whole genome, we're cutting it up into little pieces, and then we're pulling the pieces apart and then allowing them to pair up with pieces of DNA from another organism, and we're seeing how much can pair up. And if they can pair up really well, then they're probably the same species, right? So if over 70%, so make sure you can know these rules for species, okay? So 70% pairing or better means the same species. For sequencing, though, we need 97% or better to be the same species in sequencing. So those are detailed numbers that you really should know as well. The GC analysis, that was one of our things that we're using in the gram-positive bacteria. Separating them into groups, it can also be used in other ways of just kind of saying it is or it isn't this organism. It can't tell us the details of a particular species. I did go into the little mini lecture on then the groups of bacteria, so that's what you should kind of fall back to. I kind of took what would be. Now I'm going to switch over to the bigger study guide where we start to get to all these listings. So we have the proteobacteria, for example, and then I have the alpha, beta, gamma, delta, epsilon, and then there's some little things about them. So what to know about when we get to there. So pretty much everything I did in that last little lecture, that's it. That's what you would need to know. What should you even look at this table? Yes, I would. And the reason I would is I would kind of take the name of the group, like, so for example, alpha proteobacteria. Be able to tell me, are they gram-positive or gram-negative? So the proteobacteria are a gram-negative group. So you should be able to do. So that little bit. The next thing you'd be able to do is give me a characteristic. Is there any characteristic or trait associated with that group? So alpha proteobacteria, and there was one term introduced called oligotrophic. So I said that they do best where there's almost no nutrients, very low nutrient environment. So that would associate with them. The third thing you might then pull out is some of these have a list of, 2 or 3, or some like the gamma proteo. I said that's the one where almost all the bacteria that we work with in the lab belong to that particular group. You don't have to know every single one, but especially when there's a list of them like this, that you should pull out one of them that maybe you recognize, or especially if it says on the side here that it causes a disease, like the plague, which everyone has heard of. Yersinia is the Yersinia pestis. That's the bacterial species that causes the bubonic plague. And that belongs to this group of gamma proteobacteria, so maybe kind of being associated with its name could be helpful. Those, though, are the types of questions that are only going to count individually for maybe a single point on the exam. So like in a matching section that I would have, each of these would be worth 1 point. And I'd have 10 of them probably total. So I would not spend the majority of your time trying to memorize this entire table and not study the cell wall structure and be able to draw it from scratch, or not look at the parts of the flagella. Those are things that will have probably multiple questions about them, adding up to 10 points alone or more on just the membrane. I mean, I would say probably there's like 15, 20 points of the exam just related to the cell wall structure spread out among the essay parts or sketches and the multiple choice questions. So that's not something you want to brush off to try to memorize all the names on the table. These are, like I said, examples, and I'm only going to be pulling out really the name of the group and like an associated characteristic with it. A few of them you may also see in the multiple choice section as well, but again, very few. I wouldn't spend a ton of time in it. Just review the video that I just did, the lecture on the classification. gram-positive versus gram-negative, Firmicutes, actinobacteria for gram-positive, proteo, non-proteo for gram-negative, and then any little details or unique things I said about each of them in there. That's where the focus should really be of your time. And this should just be, if you have extra time, you're kind of like, well, let me pick a name or two to go with them, then that would be great to be able to do that. But I would not make that a high priority. So I usually use this table. Actually, I made this table to be used in lecture because it's hard sometimes to find anywhere that there's a good summary of the groups, of a characteristic, and of some of the members. Your book, again, does a pretty good job of this, but you have to kind of read through part after part after part to kind of gather up this kind of information. I just condensed it into one spot. So this is less of a guide to study for the exam than just a summary of material that we kind of covered with some just examples of some things that is added interest and something that I use in lecture. This again here is sort of an example. I'll have a 10 question matching like this on the exam. Some of these will probably be here. Some of these there will probably be some things in it that are not here, that are someplace else in this area. But this just gives you an idea of the types of terms I'll pull out and the types of things I'll have to associate with them. So I would definitely go through it, take a quick look at it, and if you could do it, you'll probably see a few of these will also come back up on the exam. But then I would just go through that table of identification of bacterial diversity. The last one here actually has an archaea one as well. Now I did do a separate little archaea lecture, and I just did it from the aspect of the differences between archaea and bacteria. That's all you need to know. This is actually where we actually go into archaea diversity and the subgroups of archaea. That's not here, so that's not on the exam. Do not dive into the breakdown of the two different groups of archaea and then the subgroups within them and their characteristics. They are largely broken down by metabolism, so by unique things that they can do with their metabolism. So and we're not going to really get into that, at least not at this time. It's something that will come up later when we actually talk about metabolism. We'll bring in the archaea and some of the unique things that they do later on. So for now, I would do not focus on this at all. Do not really study this at all. Just a little bit that I talked about with archaea. As far as the questions go on the short, and then back to the short study guide, I try to really abbreviate these questions. I said in this, there are, 4, 6, so there's 11 things here. Like I said, I'll probably have 8 questions on the exam, and you'll be choosing 6 of them, and they'll basically come from these. So they might be slightly reworded, but there's not going to be anything different. I guess I could say this. There will be nothing in those questions that isn't here. Okay, so if you know all this, you'll know. I might ask it in a different way. I might ask a different aspect of it or something. But if you know this, or like essentially a part of it, as opposed to the whole question, I might abbreviate it. But if you know this, then you'll be absolutely fine for that part of the exam as well. So hopefully this helps give you a little bit of direction, like I said, and look at those other 2 little final lectures on differences between bacteria and archaea, and then the overall classification of bacteria, just those terms and the groups they belong to, and any characteristic would be all you would really need for that. You're not going to have to go into depth of listing 10 organisms for each group. So that would be pretty much it. So focus on the cell structure. Again, that would be probably your best time spent best is focusing on that.